in previous video we people have understood the process of selecting transformer from non-transformer by using selectable marker, that is our antibiotic resistant gene. But the process of this, it becomes tedious and it is a cumbersome process, as this require two different plating which involves two different antibiotics. Okay, so for the process of selecting this transformants from non-transformant, we are having one more technique, and that technique is called as your blue-white screening. So apart from your selectable markers, antibiotic resistant gene, one more technique which is very easy, which is used nowadays, right, is called as your blue-white screening. Now understand properly. If I am talking about bacteria, say for example if I am talking about E. coli, तो E. coli के अंदर क्या present होता है बच्चों? Plasmid is present, yes or no? So here I have drawn plasmid of bacteria. मानो कौन सा बैक्टीरिया यहाँ पे मैंने लिया है? E. coli. Okay, so this is plasmid of E. coli bacteria. Now look, this plasmid have a specific sequence of DNA, or I can say nucleotide, right, that forms LAC-Z gene. Okay, can you see this? This segment is gene. ये gene का नाम क्या है? LAC-Z. This will be clear once you understand your chapter molecular basis of inheritance. इसमें lac operon करके theory है, वहाँ पे this LAC-Z gene is mentioned. अभी के लिए plasmid of a bacteria have LAC-Z gene. Now this LAC-Z gene, अब ये gene है, ये DNA, it undergoes what? Transcription, it undergoes translation to form enzyme called as beta galactosidase enzyme. ठीक है, this LAC-Z gene undergo transcription, translation to form protein. We very well know that all enzymes are protein in nature, right? So expression of LAC-Z gene produces an enzyme called as beta galactosidase. Normally बीटा गैलेक्टोसिडेज का फंक्शन क्या होता है? टू मेटाबोलाइज लैक्टोज प्रेजेंट इन द मीडियम. लेकिन यहाँ पे वी आर गोइंग टू ऐड अ क्रोमोजेनिक सबस्ट्रेट जिसका नाम है एक्स-गाल. इंस्टेड ऑफ लैक्टोज हमने मीडियम में क्या ऐड करा? योर क्रोमोजेनिक सबस्ट्रेट. क्रोमोजेनिक सबस्ट्रेट का मतलब द सबस्ट्रेट हु प्रोड्यूसेस कलर. ओके, क्रोमोजेनिक का मतलब क्या होता है? अ सबस्ट्रेट हु प्रोड्यूसेस कलर. ठीक है, कौन सा सबस्ट्रेट लिया है हमने यहाँ पे? एक्स-गाल. नाउ दिस बीटा गैलेक्टोसिडेज एंजाइम मेटाबोलाइजेस दिस एक्स-गाल प्रोड्यूसिंग ब्लू कलर. Hence the colony of bacteria appears blue in color. Simple process है ये. Okay, ये बैक्टीरिया में LAC-Z gene, this gene expresses to form beta galactosidase. Okay, अब मीडियम में क्या डाला हमने? एक्स-गाल. Upon metabolizing this एक्स-गाल by beta galactosidase, आपको कौन सा कलर मिलने वाला है? ब्लू कलर will be formed. Okay, now if we are placing our gene of interest, this green color portion is gene of our interest. Okay, this gene of interest we have added where? As you can see over here, where we have added, where your gene LAC-Z is present. मतलब recognition site of particular enzyme will be somewhat over here. यहाँ पे होगी recognition site. Okay, so we have make cut. हमने LAC-Z gene काटा और ये आपका जो gene of interest है by use of DNA ligase हमने attach करा. Now due to addition of this new gene of our interest, LAC-Z gene gets inactivated, जिसको आप क्या बोलो? इंसर्शनल इनएक्टिवेशन, मैंने आपको कल पढ़ाया, ओके, इंसर्शनल इनएक्टिवेशन, नया आया पुराना इनएक्टिवेट हुआ। नया कौन आया यहाँ पे, ये जीन ऑफ इंटरेस्ट, जिसकी वजह से लैक्सीजीन बिकम्स inactive, इसको बोला जाता है insertional inactivation. Now as the gene is not active, there will be no expression, no formation of beta galactosidase. If you are going to add X-gal, then ये एंजाइम ही नहीं है तो ये मेटाबोलाइज होगा नहीं होगा. If this is not metabolized, there will be no formation of any color. So this colonies appears white in color. ये जो कॉलोनी है इज गोइंग टू अपीयर व्हाइट इन कलर. नाउ प्रोसेस एकदम सिंपल हो गई. इफ यू आर गोइंग टू कीप योर बैक्टीरियल कल्चर इन अ पेट्री प्लेट कंटेनिंग द न्यूट्रिएंट मीडियम, ये क्या है बेटा जी आपका न्यूट्रिएंट मीडियम, अगार प्लेटिंग, ओके, अगार प्लेट भी बोल सकते हो आप. इसमें मैंने क्या डाला, कल्चर डाला, राइट. कल्चर मतलब वी हैव एडेड योर बैक्टीरिया, द बैक्टीरिया विच अपीयर्स ब्लू इन कलर, which appears blue in color, okay, वो कैसे हो गया आपके नॉन रिकम्बिनेंस, ओके, नॉन रिकम्बिनेंस इन द सेंस यहाँ पे कोई इंसर्ट नहीं हुआ, बट द colonies who appears white in color, यहाँ पे मैंने ग्रीन पोर्शन दिखाया है, who appears white in color, they are our recombinants. So by process of this you can easily separate recombinants from our non recombinants. तो ये क्या हो गया आपका blue white screening. This is very important one, okay, it based on production of color, okay, it based on addition of your chromogenic substrate, simple as that. Let us now understand vectors which we are going to use for cloning genes in plants and animals, okay. So far we people have understood properties of cloning vector, features of cloning vector, and how we select transformer from non-transformer, right. Now we are going to understand vectors for cloning genes in plants and animals. Two such example you people have to memorize. One I have written a bacteria, gram negative bacteria called as Agrobacterium tumefaciens. This is pathogenic bacteria which is responsible for causing crown gall disease, okay. This Agrobacterium tumefaciens is responsible for causing crown gall disease in several dicot plant, okay. And second you people have to memorize your retroviruses. So have a look what I have written about Agrobacterium tumefaciens. So once again आपका Agrobacterium tumefaciens ग्राम नेगेटिव बैक्टीरिया है, पैथोजन ऑफ सेवरल डाइकॉट प्लांट्स. पैथोजन इन द सेंस दे आर रिस्पॉन्सिबल फॉर कॉजिंग डिजीज. नाउ लुक हियर, इफ आई एम टॉकिंग अबाउट दिस Agrobacterium tumefaciens, तो इसके अंदर जो प्लाज्मिड होता है, द नेम ऑफ प्लाज्मिड इज TI प्लाज्मिड. In that TI plasmid there is particular gene जिसको आप बोलो tDNA, उसको क्या कहा जाता है tDNA. Once this tDNA enters through cut portion of root or stem, it converts normal plant cell into cancerous cell. In the sense यहाँ पे they are responsible for causing tumor. They are responsible for causing tumor. Once again what I said, your Agrobacterium tumefaciens have a plasmid. This plasmid is called as your TI plasmid, okay. There is specific sequence of gene which is present over here which is called as your tDNA. Once this tDNA enters your plant cell, it converts normal plant cell into cancerous cell responsible for causing tumor. Similarly, if we are talking about this retroviruses, to be more specific, this in case of your animals and this in case of your plants, okay. So retroviruses in animals, again they have ability to convert normal cell into cancerous cell. So what I have written in black line, let's understand a better understanding of art of delivering genes by pathogen in eukaryotic host has generated knowledge to transform these tools of pathogens into useful vectors for delivering gene of interest to human. Simple सा मतलब है, we people have understood their art of delivery, how they are responsible for causing tumor, and we people have modified that. We people have modified that to deliver gene of our interest. Simple as that. ये दोनों हमने proper study करा, right? And we use how to deliver this gene of interest in humans. Okay. Now, if I want to explain in diagrammatic manner, तो ये क्या है आपका एग्रोबैक्टीरियम ट्यूबिफिशियन, ये उसका नॉर्मल डीएनए, दिस इज योर plasmid, this plasmid is called as your Ti plasmid, black color portion represents your tDNA. This tDNA is responsible for changing normal cell into tumorous cell or cancerous cell. Now what we have done, जैसे मैंने आपको अभी यहाँ पे बोला, we have modified this Ti plasmid to deliver gene of our interest. Okay, so have a look, this is your gene of interest. जो भी ट्रेट आपको चाहिए उसका ये जीन है. This gene we have inserted inside your tDNA, as you can see over here, the green color portion you can see over here. So this represents your step number 1, that is insertion of gene into plasmid using of course restriction enzyme, then joining by DNA ligase. This forms your recombinant Ti plasmid. ये क्या बना आपका recombinant Ti plasmid. Okay, now this tDNA becomes inactive. आप इसको क्या बोल सकते हो, insertional inactivation. So now this tDNA do not cause cancer in plant cell, rather it delivers our gene of interest. Okay, so once again यहाँ पे tDNA जहाँ पे है वहाँ पे restriction enzyme use करी and we have added our gene of interest. ये आपका क्या रेडी हो गया, recombinant Ti plasmid. Now this recombinant Ti plasmid we are going to introduce step number 2. We are going to introduce in a plant culture. Simple as that. So introduction into plant cells in culture. तो ये क्या है बेटा जी आपका plant cell, and मानो this is the chromosome, right? And to this chromosome your tDNA carrying new gene is introduced. Okay, so this is your plant cell. ये portion मैंने zoom out करा. What I have written, this is tDNA carrying new gene within plant chromosome. Simple as that. Now this plant grows with new trait. So this is your point number 3. The plant grows with new trait, right? And this agrobacterium now delivers gene of our interest. They are not responsible for causing Crohn gall disease. Okay, in short, they are not responsible for causing tumor. Okay, so this is all about your cloning vector. I hope you people understood it well. Once again, इसमें दो चीजें आपको याद रखनी है, Agrobacterium tumefacium and your retroviruses. We have understood art of delivering genes, right? We have modified that particular segment and now we are using the same for delivering our gene of interest. Thank you.